Welcome everyone to another episode of the Evolutionary Hologenomics Podcast. Today, our guest is Jacob Rasmussen from Copenhagen University. He's a PhD student, and it's so great to have you. Jacob, thank you very much for joining. Will you introduce yourself in a few words? Just who are you? What do you do at the center? And what are your research interests?
Yes, thank you, Anna. My name is Jacob. I'm a PhD student in Morten Limborg's group called Applied Hologenomics. I'm mostly working on interactions between bacteria and its host in salmonids. And that's basically what I've been doing for 3 years now.
That's a very short introduction to summarize lots of work that you actually do. So you made that like the one-line punch. But
that's good.
Today we will unpackage a little bit of the work that you have been doing. And for those who follow the podcast and who have been listening to previous episodes, usually I just sit down and have a chat with the various people from the center and see what they're up to with their research and what it all means. But a big part of doing research is actually to go to conferences and present it, as well as discuss with other scientists, collaborate with other scientists, and of course, publish your results in peer-reviewed journals. So Jacob recently published 2 papers. And so I thought that for this episode today, we were going to do a little bit of a journal club. So I'm going to just start by letting you know in very short, what is this scientific paper thing, because the non-scientist audience might not be familiar. And then we dive into the actual papers. So basically, the publication is written by the people who do the work, and then it's submitted to journals that have reviewers to make sure that the science is sound and it makes sense and it was done ethically and so on and so on. So there is usually a title, and then there's an abstract, which is basically a summary, so that people can actually scan and see if this paper is interesting to read or not. And then there's an introduction, which is basically a mini literature review with all the sources, because we're scientists, so we cite the sources. We're not just coming up with stuff, hopefully. Then materials and methods, so what exactly has been done. And this part sometimes can come at the end as well, depends on the journal. And then finally, there are the results, which is kind of the heart of the paper. After doing all the work, what came out of it? And then there's a discussion part, which is in the light of what is known or the hypothesis, what does it all mean? And finally, we have acknowledgements or conflict of interest sometimes, and then the whole entire list of all the references throughout the paper. So that was kind of in short, just to introduce all this. And now we get to dive into the first of 2 papers that Jacob has recently published. And first of all, I think we need to unpackage this title. There's a lot of, it's a mouthful and there's a lot of complicated words here, but I promise you, we will make it simple for you. So I will read it, and then maybe Jacob, you can unpack the little bits. So it's called a Multiomics Approach. So what is this multiomics thing? Unravels metagenomic and metabolic alterations. So what are these metagenomic and metabolic alterations of probiotic and symbiotic additives? So what are probiotics and symbiotics in rainbow trout? I think that part we understand. So it is something that happens in rainbow trouts. Jacob, let us know in short, what does this all mean?
So in short, that's mostly difficult, right? So first of all, I think to explain what multiomics is, we should maybe explain what omics is. Omics is basically a word we use for huge data sets, like vast data sets, which are normally in the realm of genes, which could be like all genetic material in a person. That would be the genome, right? And if you are investigating the genome, you are doing genomics. Yeah. And the same could be for all the RNA which has been encoded by the genes. That would be transcriptomics, because we are working with the transcripts of the genes. And then you could do like all the proteins which has been expressed by the RNA and synthesized. So that would be proteomics. And lastly, you normally also do something called metabolomics, which are investigating all the metabolites in a given sample. And metabolites are normally characterized by being molecules below 500 kilodalton or something like that, I think. Yeah. So in this case, we use an approach which use really big data sets, and we use multiple of them. So that's the part of the multiomic. And what we use it for is to like analyze the metagenomic variation in our study. And the metagenomic variation, that's basically when you have multiple genomes, so multiple organisms, genomic content. Yeah. And so we investigated the metagenomic and metabolic variation when we used something called a probiotic and the synbiotic. And a probiotic, we all know, that's basically what do you say? Some kind of diet where you have added like normally health beneficial bacteria, which are basically giving some kind of beneficial effect. But sometimes probiotics itself is not enough. Like the bacteria is not enough itself. It might have a struggle cultivating in the host, or in a gut, or in any kind of surface, basically, right? So it would also need some kind of fiber, which would be a prebiotic. And when you add both a prebiotic and a probiotic together, we call it a synbiotic. Yeah. So here we investigated a diet with additive bacteria, and the same diet with an additive bacteria and with some kind of fiber, to see how this basically affect the bacteria in the gut and the whole metabolic and all metabolites in the rainbow trout and its intestine. Did that make sense in a very short way?
Yeah, thank you very much. That was a lot there. So basically, you studied rainbow trout and fed it with some probiotics or symbiotics. And then you had a look at all these different layers of genes and proteins and metabolites and all these things, and see how all this affects the health and growth of the rainbow trout. So what is the importance in all this? Because obviously there's a big industry of farming rainbow trout and Atlantic salmon and other species, and it can be even not just species on the water, but also on land. So what is the advantage and the insights that such studies can bring us?
Yeah. So first of all, like in agriculture, like when people have to like test a new diet composition or feed in general, they do like feed the fish, see how it goes, right? do they perform better? Sometimes they do, sometimes they don't. But actually, everything in between, like, it's a black box. So we try to basically unravel this black box and see how is all these different data layers, how are they affected when you add some kind of diet, and especially how is the interplay between bacteria and rainbow trout in this case affected. This can be extremely important to basically direct your research and development in like a feed industry, for agriculture, or for basically any kind of what's called live production animal, right? So yeah.
So it's a mechanistic study. So it's not just, you put something in there, you get something out, but you have no idea what happens in between, but actually you look into the mechanism and to the quantifications and what's what exactly. So then the diet and the benefits can be a lot more tailored. So that's very interesting. And of course, what I want to know now is, so what did you find? But before we go into that, could you tell us just briefly what was the experimental design? So to feed these different, the rainbow trout with these different probiotics, and then how did you collect your samples and what did you do with them? Very briefly.
Very briefly. So basically we had 3 different kind of diets in this study. We had a diet with no additives, and then we had a diet with a bacterial additive, and then lastly we had a third diet with a bacterial additive and a what's it called, a fiber. So these rainbow trout had been fed for 29 days, and after that we slaughter them and we dissect out the whole gut, and then we take the gut content and the mucus layers in the gut and store that for later processing, where we then basically extract the DNA for bacterial composition, and we also extract the metabolites in this study, and analyze these 2 things basically.
Yep. Again, very simple explanation, but I know it is a lot of work and a lot of samples, and also then at the end a lot of bioinformatics to to go with analyzing all the different layers. And so I guess your main aim was to have a look at the sort of the micro scale, but I guess you also, like, one of the first things that you do when you sample the animal so that then you can actually make correlations is to actually look at the classic weight and the efficiency of growth and all that. So what did you observe with the different diets?
I'll just like go to one of the first things, because in like classic feed performance analysis would be to look at like how does a fish perform according to like gain of weight. So how much does it grow during this feed trial? And this second thing would be how much muscle is transformed from 1 kilo of feed to 1 kilo of pure meat, or gross weight, right? And then the same we do for lipid efficiency. So like how much of the lipid has been actually has been taken up by the fish, and the same with protein. Does that make sense?
Yep.
So in this case, we did these 4 measurements of gain of weight, feed efficiency, and lipid efficiency, and protein efficiency.
Right, and there were differences with the different diets?
Yeah. In general, we didn't see huge differences in gain of weight, and that's quite common, actually. But there was a trend that the control in this study did insignificantly better, you would say, in a scientific way, right? But there's a trend. And the same with the feed efficiency. The interesting thing was that in the nutrient profiles, we saw that there was a significantly higher efficiency when you added probiotics in lipid uptake, and the opposite in protein uptake, where we saw that the control were significantly higher than both pro and symbiotic, which could seem like if you add a bacteria, then you can basically modulate the nutrient uptake of the rainbow trout.
Yeah.
Though this has no significant effect on the growth weight. But
yeah, so already here with this very basic observation, we can already see that if you just look at that, then you're missing out on the big picture and then actually what is happening inside at sort of the molecular and interactions level. And so, but the main goal here was also to obviously find out then what kind of different bacteria were there and what they were doing. So was there a difference in what kind of bacteria you could find with the different feeds?
Yeah, there was. So this is a very, though we should not talk about the figures, but in front of us, for people who are seeing this, like it's a very complex figure. But we did see very, very different microbiomes or microbiota compositions. And in general, we were quite surprised because we figured out that the rainbow trout were, when you have what we could maybe call some kind of naive system, like in the control diet, it was highly dominated by one bacteria. But when we added these different diets, suddenly this dominance disappeared and other opportunistic bacteria, most likely opportunistic bacteria, came in. So there was a huge shift in the microbiota.
And just out of curiosity, so these bacteria that you added, I guess they're all deemed beneficial somehow. I guess otherwise you wouldn't be adding them.
Yeah, at least it has been beneficial in other life production animals. And of course, this bacteria is called Pediococcus. It's a lactic acid bacteria. So in general, there's a consensus in like these functional feed industries that lactic acid bacteria are beneficial for higher vertebrates. I cannot really discuss that because I'm not an expert in that. But yeah,
it has been described that they have benefits, and so when you add them, this dominance of mycoplasma that exists when you don't add anything just is disrupted. And does that mean anything in particular for the health or growth of the salmon and trout? Do we know? From
this trial, there was no significant change, right? But there was a trend that they had a lower what's called efficiency ratio in proteins, right? So it might be something that mycoplasma could help, like in protein uptake, because it also affected the feed efficiency ratio, but not significantly. So it's quite difficult to tell, actually. I would still say.
Yeah, but nonetheless, the observation is there that indeed the composition does change. And then you also mentioned that you had a look at different mechanisms, different molecules and metabolites. So what is the landscape of that?
Yeah, so like this is like the first thing you would investigate, right? You see by a glance that there is a compositional difference in bacteria, but so what? Like it might not mean anything really. But what we first did in this case, we investigated just the presence of bacteria. And then secondly, we investigated the presence of, we made an investigation of which functions are present, bacterial functions are present in the gut. And for this study, we found a huge difference in specific bacterial genes, which are mostly secondary metabolites, these isoprenoid biosynthesis genes, which we really don't have a clue about what they're doing, because science in general don't really have a clue about what they are in higher vertebrates and how this affects the interplay. One thing we found was the arginine biosynthesis was highly altered also, which is a part of protein synthesis. And in general, these proteins are essential to salmonids, is at least my belief. So, and so it might be a beneficial thing. And we also saw that I have called it polymer utilization, but it's basically degradation of complex fibrils like chitin, which is normally found in the exoskeleton of insects and crustaceans. We found that all these genes were following the presence of mycoplasma, this dominant bacteria. So if mycoplasma was dominant, there was a high amount of these genes, and if mycoplasma was missing, there was nearly none of these genes. Yeah. So first of all, we found that that the composition of bacteria might affect, at least on a genetic level, the presence of these genes, which are secondary metabolites and protein amino acid stuff, and also carbohydrate utilization, right? But to really actually validate if they do anything, right, you need to actually measure presence, absence, or abundances of these metabolites, which are amino acids or secondary metabolites or degraded carbohydrates or all kinds of these things, right?
Yeah, and so in this study, you did not do that. You basically, so that people can understand, because there's a lot of things people actually measure in the lab. So if we're talking about people who only do wet lab work, although nowadays there's a lot of analysis going on as well, there's a lot of in silico work. So in the computer, and there's a lot of databases and a lot of things that basically your results can be compared with and then mapped with things, so that you can try and figure out by approximation what's there and the different pathways and what can be happening. So when you say that, so this could be a next step, right, to actually measure these, because now you have the expression, but you don't have the result of that expression, which would be the actual protein or the metabolites.
Yeah, actually we did that in this study, which is the sweet part of this study. We validate, in the end, basically how the metabolic profiles, the metabolic alteration is behaving in the same way as the microbiome composition and the microbiome genetic composition also. So actually, that's, if I can. That's actually what we see on the screen here. Sorry to people who are listening, but we actually try to validate these things on measured metabolites, and just to explain, we found 24,000 metabolites or something like that, and that's a lot. Or actually, we cannot call them metabolites. Chemists would normally be very angry about that, because we don't really know if they are metabolites or any other kind of noise when we use these fancy instruments. So we call it features. And what you would then do is in order to figure out if it is a metabolite, we would normally like with DNA, we normally blast it towards a database, right? And then we will see that the DNA has the same sequence composition as a Labrador or something like that, right? But here we cannot do that. So we do a lot of machine learning. So we make classifications on the metabolites based on all the mass charges and substructural what's called substructural features, and then we can see that, hey, this might be an amino acid or peptide or bile acid or a secondary metabolite like terpenoids, or like it could have like sugar, a lot of hydroxy groups, so it could be like sugar molecules, right? But in general, we saw here that all the metabolites overall the composition is following the bacterial composition also. And we also saw if we got down on classes of different metabolites, we also saw the same trend for amino acids and these secondary metabolites and also bile acids. So it really actually gives us a clear idea that it's not only the genetic composition which are affected by these different feed additives, but also the overall metabolic product machinery in the host itself, but also in the bacteria.
Yeah, that's really great to see, because then it makes the study a lot more robust, to see that in theory you have this gene and then you have this message, and if you just stop there, then the message is the potential, but a million different things can happen in between. So you went and actually measured the products, and they do correspond to what the last step was indicating, which is really, really great. And I think this is before going into the conclusions, you touched upon something that I think it's very important. When you said that just because there's a difference in composition in the bacteria, it may not mean anything, because in ecology, to kind of, and this is valid with genes as well, to kind of ensure that something very important does go ahead no matter what. Usually there's a lot of redundancy. So in terms of species or genes, or so to make sure that if something fails somewhere along the way, it can be that even though it's different in the middle, the result can still be kind of the same because of these compensatory mechanisms. So I think that's an important thing as well.
Exactly. And that's where the power of metabolites basically comes in, because metabolites are most likely the closest we can come to like a molecular phenotype, because it's literally the product of proteins and enzymes. So at least in my realm, I would say it's the closest to a molecular phenotype we can go. Yeah.
So this is a very neat kind of proof of concept, if I can call it that, an insight into what this kind of study can unlock, and how it can help the industry. So if you have a few kind of main thoughts and conclusions in that direction, what would you say are the main conclusions and main sort of achievements from this paper, this study?
So yeah, this whole paper is, as you say, it's mainly like a way to make a framework on how to investigate this black box. And in general, I would say that the conclusion is that you cannot, though we see that the different layers actually complement each other and they follow each other, you cannot always rely on that, right? So you really need to investigate multiple layers to see not only the gene composition, but also the functional composition and the phenotypic variation, right? And I guess that's the main goal of the article. Of course, we also saw some differences, like that the change of diet also changed the bacteria composition, but that's really not a huge surprise, at least not if you read the literature. But I would say,
Yeah, so scale and details are very important, and I think I know for a lot of projects in the Center for Evolutionary Hologenomics where we work. So for example, we have this big European project called 3D-omics. And again, it's all about the scale. And we, in this project, we are going as far as actually trying to map everything in the 3-dimensional space, because not only what is there, but where they are and what are they doing is very important to see the whole picture, rather than just putting a section in a blender and say, well, this one has more and this one has less, or this one has 5, these are the species and this one has different species. But then, so what does it all mean? So it's good to see that a lot of very different, but with the same spirit, projects are being done here at the center. And so, but you have 2 papers, because one is not enough, obviously. And so this one is about, but all of them, and I think the overarching narrative here is the importance that the role that the microbiome plays in the health and other aspects of the host, and then the dynamic and the interaction between the two by looking closely into what they're doing. So this paper is about health and growth in general, but then of course another big area and concern, and something that the aquaculture industry is very aware of and always trying to improve, is obviously also when things go wrong and when the animals get sick. And so your second paper on this subject is more into looking at when things don't go so well. And I think maybe we can also try to start by unpacking the title. It's another mouthful. Yeah. So it's called Integrative Analysis of Probiotics, which by now we know what they are. Pathogenic infections and host immune response highlight the importance of gut microbiota in understanding disease recovery in rainbow trout. This one is maybe a little bit easier to unpack. Go ahead.
Yeah, so now we know what probiotics are, right? So that's like when you add a bacterial component, mostly like known as beneficial, into a diet. Then you have pathogenic infections. Actually, I wouldn't say it's a mistake that it's plural, but it's only one pathogen in this case, at least from us. So yeah, that's when you infect a pathogen. And then the host immune response is, in this case, we looked at immune gene related as a like expression of immune genes, immune related genes, as a response when we did this trial. And then of course the gut microbiota, that's all the present bacteria in the gut of the rainbow trout. So yeah, basically this means that we had a lot of data and we did a full analysis on a lot of different data where we accounted for a lot of different things, like presence of probiotic, presence of bacterial pathogen, and then different immune gene related genes also, and saw how this affect different bacteria in the gut of rainbow trout.
Yeah, and so as before, I'll ask you just to describe a little bit in as a summary, kind of the experimental design that you had for this this trial.
Yeah. So in this trial, we narrowed down the diet, but instead we basically made the fish sick with something called Yersinia rocari, which is something called it's a causative agent of enteric mouth disease, red mouth disease, which is very common in agriculture and a very huge problem sometimes in agriculture when it goes rampage, basically. Not the fish, but the bacteria. So what we did, we had two different diets, and then we basically gave we tried to give both groups of fish. We challenged, that's the word. Sorry, we challenged the different groups of diets with fish fed on different diets with this Yersinia rocari, and saw how did this affect survival and how did it affect microbiome composition and these immune-related gene assays and all these things. And in general, we saw that on the survival we didn't see any differences, but it would also have been like quite surprisingly, because this pathogen is really a harsh one. Yeah.
So basically, if the fish get it, they die. It's not like they get sick. Mostly they die. How deadly is this?
So it's around 50%. So of course we cannot say that the last 50% had it, but we can say that of all the fish we sample, like, so the thing is, like, in these trials you are not allowed to let a fish die from a pathogenic infection because that's unethical. Yeah. Right? So I would say it comes back to doses, right? We want to let them become sick without dying of it, because that's unethical, of course. So yeah, but in nature, they would die in general.
Yeah. So you have data to kind of infer that from this point on, then they would die. And just for people to have an idea as well, every time there is experiments with animals, there's a lot of paperwork, and there's an ethical committee that need to approve your experiments, and you need to prove that it's absolutely necessary, and you have to use the minimum amount of animals that will give you your statistically significant results. So there's vets involved and other people to make sure that the animals don't suffer, and you have to sort of sacrifice them beyond or from a certain point to avoid further suffering. So that's important to highlight, because it's not a discussion, or it's not very widespread how these things work, but it's important to highlight that all of these mechanisms are in place, and there's very strict laws to make sure that the animals are protected. And so, because in our center, a lot of the times we, with aquacultures, we have facilities where the animals are out in the ocean, and they have these huge tanks, but they are in the ocean. But this one, where you actually infected them with something that is not so good for them, was this done indoors, or where was the setting?
This was all done in a RAS system, like a recycled agriculture system. So a land-based unit. I don't know if that's common, like, but I would think it is fairly common, especially because it's normally done on small juvenile individuals. Mostly because it's expensive to have what is it called, test animal living for 3 years with no reason, but also because it's the most critical period for a lot of these farmed animals. So yeah.
Yeah, and so what else did you observe? So you treated them with this pathogen, and then they were on different diets. And the goal here was to, I guess, first of all, have a look at the microbiome. There's a trend here of kind of like step one, we want to look at this, then step two, and then we get to the actual purpose of the different studies. But there's a few set of basic steps that are common to all of them, I guess.
Yeah. So the first thing we did was, of course, to observe if there was any survival differences, and there was not. As again I said, it would have been a very big surprise if you could magically feed a fish more healthy against enteric bacteria pathogen. That would have been very cool, but also highly improbable. And then the next thing we wanted to test is, okay, the presence of these pathogenic bacteria inside the rainbow trout, to see if was this pathogen everywhere, or was it only in infected fish, because that's a quite good control to have. And we found it only in the infected fish. And then the next thing would be to investigate how the microbiome composition is. And in this case, we saw that as the previous paper, that the control diet without infection had a dominance of mycoplasma, whereas the composition in the fish without infection but fed with probiotic, it was of course changed to another composition, as we saw in the previous paper, right? But what we saw with both of them was that and mostly with the control diet, is that there was a huge shift between infected and non-infected individuals. So there was a huge shift for control, but not as big of a shift for the probiotic fed individuals.
Right. And so these observations, and it's always good when you do a study and you see different things, then how helpful can this be, or what kind of indications can they provide?
Like
for example, the animal production industry, in terms of what is the role of the microbiome and these feeds when animals are actually not doing so well.
So one of the things is that in general, it's believed that when you have a pathogen coming in, like as an enteric pathogen, so it comes through the intestine, is that you would like it to not change the microbiome, because then the recovery would be more stable, most likely, because there's like what can you say, like there's not been this dysbiosis in the gut microbiome or in the gut community, so it would be easier to go back to how it was afterwards of the infection, right? If that makes sense. So these kind of compositional changes could be of quite great value to evaluate if the diet is better for the gut microbiome during infection or not.
Yeah. And so I guess as a parallel with the previous paper, we do a treatment, we see the effect, we have a look at the microbiome composition, and then did you go further to then have a look again at the different molecules and gene expression and what that might mean for the host?
I would love to have done that, but in general, as it is with a lot of these studies, they are fairly expensive, and we didn't really plan to make this kind of this part of the trial as comprehensive as the first part. So we didn't go further deep into molecular layers, but what we did was we wanted to investigate the host responses. So in this case, we looked into like different immune gene related genes and how they were affected. And in general, we actually saw an overall trend that not all of them were significant, but in general, there was a trend that there was a lower expression in post-infection in individuals fed with probiotic. So in some way, there's like a lower gene expression of all these immune-related genes when they are fed on probiotics, indicating that it could be like higher tolerance, or that's something I really don't know, because I'm not an immunologist, and that's really a complex matter. So, but yeah,
but it is a start, and a lot of health-related studies, whether it's fish or people or something else. There is a known battery of immune genes nowadays that people test for, and then they see what's going up or down, and then what that might mean in terms of not only the immune response itself, if it's better or worse, and then of course there is the effect of that if the body can somehow respond better, then the infection is not going to be as bad. So even though these are trends, they seem to go in a specific direction. And as with many studies, especially the pilot studies, it's always good to see this as a base. But at least I think with these two studies, the common feature here is that they do seem to both highlight the importance of doing more of this and investigating a little bit in more detail.
Because if we haven't done this, like I will talk a little bit about it in a second, but if we haven't done this, the study has been done right, we saw differences, and so what, right? Like we saw differences in gut microbiota, and that could not, then that's it, right? But it's not surprisingly that you have sick fish, healthy fish, and there is a difference in gut microbiota. It has been seen quite sometimes. So one of the things we did was, because we had all these genes, it was to see if we could like find correlations between regulation of these genes and specific bacteria. But in order to find specific bacteria, we needed to like find biomarkers between fish, healthy and what's infected and non-infected fish. And we did that with both of the control and the probe. And in general, we saw with the control, it was easier to predict. There was a way higher amount of true positives than false positives, because the biological signal was simply way clearer, because the control gut microbiome was simply way harder affected. So yeah, but here we saw that the presence of Pseudomonas, as an example, suddenly came in when fish were infected. It was what we call prevalent to infected fish. So it was mostly in infected fish, and it was also classified as a biomarker for infection. Whereas we had two others like Mycoplasma and an unknown Enterobacteria CA called Endosymbiont 8, very classical bacterial name, which were actually prevalent to non-infected fish. So they were biomarkers for health or non-infected fish. They were mainly present in non-infected. And actually Endosymbiont 8 was also, we saw the same trend for that one in probiotic fed individuals. So we took like a lot of these different candidate biomarkers and narrowed down the whole microbiome to only these different bacteria, I think it's 5 candidates, and looked on how was these 5 candidates affected by the presence of probiotic, the presence of pathogen, and then all these different immune genes, right? To see if we could see if some of the genes were like correlated and then we could say associated with like the specific bacteria. And it actually went fairly well. We saw that also, as the first study showed, that addition of this bacteria called Pediococcus acidilacticity, that's the probiotic, affects the microbiome. But we also saw that the infection stage, so like if it's infected or not, also does it. And then we went into specific genes, and we saw that as an example, this mycoplasma were highly affected by the innate immune system, like lysozyme as an example, which is a very non-specific immune, what's it called, immune agent, right? And the same with other bacteria also. So what we suspect is that when you have these kind of infections, you would have an innate immune system, which would basically be very like non-specific, right? And it would just rule out all bacteria in the gut, and then opportunistic bacteria will recolonize, and of course the fastest one will win, or at least the ones which are best to get the nutrients fastest would also win. So that's at least one of our hypotheses according to this.
Yeah, so both of these studies are very, very cool, and I think they have kind of a twofold advantage or usefulness, which is obviously a closer insight into mechanisms, and then the direct consequence of understanding the mechanisms then is to obviously know what to feed the animals with or how to treat them or so on based on that. And another thing is, now that you mentioned biomarkers, so this is biomarkers is something that has been around for a long, long time. And when I used to do science, we used to look at biomarkers in terms of specific genes you could test for example, like BRCA1 for cancer or whatever, because if we know that mechanistically they are, or probably in terms of probability, they are associated with a certain disease or state, then we can actually develop a battery of tests, and we can target those and measure them. And from there, we can kind of assess the health status or whatever it is that we're trying to measure. So it's great for many reasons to know the mechanism, but then there's an added bonus that then we can develop some sort of diagnostic that then helps us. So, and I could imagine this in the context of, for example, an aquaculture facility. If they had these tests that they could just pick a few. So because obviously they measure the various chemical properties of the water and oxygen, and they have to, if it's salmon or trout, depending on if it's indoors or mixed, when the salmon has to smolt and move from, change the chlorine cells and move from freshwater to, so all of these things, there's so many steps and so many quality control that needs to happen in the entire process of going from a baby until you can actually have a commercial salmon or trout. So in terms of health, I would say this is very, very important and super, super helpful for the industry.
Definitely. And like one of, at least to my surprise, was that it's not a surprise in itself that of course when you feed and rainbow trout in this case differently, of course there will also be different bacteria in the gut, right? So of course you also need to take account for that when you do like your biomarker search. So if you had this test, right, you also needed to know what kind of diet is in there, right? But in this case, we saw that there were two really good candidates for showing healthy animal or non-infected, and like Pseudomonas in this case for infection. So for both diet types, and it would be ideal to like find bacteria which are present in the fish always, right? On not always, no matter diet, as an example, right? Because then you wouldn't care about these things, but it would be robust according to the infection. But that would need way more studies, because we need to test way more diets and then you would do a huge meta-analysis on the end, right? On all these different data sets, which is also difficult.
Yeah. But again, it also highlights, as always, the context and what's there. So there's so many things in terms of water quality and the genetics of the host, and what in the feed and the microbiome and kind of the base microbiome, and then how it changes and why. That definitely we need micro scale studies to understand the big picture, so that then any test or anything that is developed needs to be in the light of all these, and we need to know for the specific tank exactly all the details, so that we can then interpret the results, because otherwise it can be completely the opposite or just not accurate.
Definitely. But yeah, and again, biomarker, it comes really down to your research goal, right? Because biomarkers are, of course, classified as biomarkers according to a specific thing, right? Like in this case, it's for infection, but if you're not interested in infection, you should use that one, right? So it's always like, depending on if you want to do targeted research, right? Or if you wanted to do this kind of, as the first paper, more holistic approaches, right? Where you just basically spray out all the data and see what is present there, or if you go for more deterministic and targeted approach, I would say, right? So, but I'm pretty like bacterial biomarkers will really, it can really offer a lot of things for industry and researchers in general.
Definitely, and you're on the right path with this. So I guess from this last paper, we know that probiotics influence microbiota composition and host immune response from your data. And then there is the importance of these, as you just said, these bacterial biomarkers that can understand the interactions between host and microbiome, which is so important. Do you have any final other remarks or conclusions regarding either this paper or both of them as a whole? Because I think it makes a nice story, both of them together.
Yeah, well, without like unraveling all kind of new stuff. This was like when we started investigating the salmonids in general, we became super surprised about the simplicity of the gut microbiome, actually, which has been quite a journey, at least for me, because in my opinion, there's more or less a huge consensus in this field that there's always like billions or millions of different bacteria in the gut, right? And when we started this research, we didn't see that at all, which were a huge surprise to me, because I was basically born in the paradigm of having these millions of bacteria in the gut. But apparently it's not the same when we see like cold water living salmonids. And that's a huge other topic we can take another day, I think, yes, because it's also, I think it's very new. Of course, it's seen in other animals, but it's not something that has been touched so much upon in the research area yet. But it's something I would love to look more into later on. But
yeah. So more is not always better. If they're happy and healthy with low diversity, maybe that's what it needs to happen. And then if there's this, as we saw from some of your results, when there's a decrease in mycoplasma, and then all of these opportunistic or other things come up, but maybe it's not as good. And actually, in when we had the episode with Raphael about the microbiome of pouch in marsupials, it was very interesting to see how the composition in microbiome changes, but also the diversity. And actually, it's a lot lower when there's a baby inside the pouch. So it's all about function. And if it works for you, that actually lower is better, then
at least in fish, it seems to be balanced, right? As long as it's in balance, right? I would also a lot of data shows some kind of consensus that at least salmonids are extremely good at nutrient uptake themselves because they are carnivorous, so they eat protein and fat, and the normal body can easily absorb that itself. So the microbiome might actually be more important for immune maturation and cofactor vitamins and all these things. So it's not that they're not essential, but it's just in the nutrient matter, it might be that the bacteria are not the first, it's not the key factor always, which is also quite surprising to us. But
but maybe it is related to that, because when we look at the digestive tract of different species according to their diet, then if you have herbivores and animals that eat a lot of fiber and all this stuff, then they might need more help than carnivores, and definitely different.
Totally. If you compare the gut lumen of a cow, right, which are what's called ruminant. Of course, they are living off grass, hard fiber, cellulose. Cellulose is not something mammals in general can degrade themselves, right? It's quite difficult. So bacteria, of course, is an answer there. But for a fish like salmon, it might not be the same thing because it's fat and proteins, right? At least in the adult stage. Of course, when they're juvenile, they would mostly live off insects. And that's quite funny, according to the mycoplasma that we found. These chitin degrading genes, which could suggest that they are beneficial in the juvenile stage, because chitin is a complex polymer in the cuticula, in the exoskeleton of insects. So that could be quite funny to look further into, but then we need more samples.
As always. But is there any study done on that kind of how the gut microbiome changes depending on the developmental stage, which obviously not only is the diet will be different, but then especially in fish, where they have to go between salt water or fresh water and then salt water, a lot of changes happen in the organism as a whole.
Yeah, definitely. So the whole intestine and kidney changes from a salmon, right? When they do multiplication, going from fresh water to salt water. So that would, of course, affect the gut microbiota, unless they are living inside literally inside the host, right? And I don't know if it's not a major aspect in the field that like doing temporal sampling, but I know that there's a lot of research projects looking into that, because it is a huge thing. Like this smoltification stage, like going from freshwater to saltwater, is a very, very critical period in the lifespan of salmons, because it's such a hard period on the host itself, because you really need to change your kidney and your intestine. So it takes quite a lot of the host itself. But as I said, I know that people in the field are actually already looking into it, but it's simply too novel.
Yeah, so one day we will know, and I guess it keeps you on the job, because there's always more and more questions, and then there's things that are more in the realms of basic science, and then of course we tend to see a bigger investment in the applied things, because the stakeholders and the interests are different. But a lot of the times also just understanding how things work from basic science then gives insights into the more applied field. And so with all these questions, what is the next step for you? What is the thing that you would like to go and study next?
So I have worked a lot in the applied part, which I like a lot. But some of these questions are also, like the biologically basal questions, are also needed to be explained in like a more wild, what can you say, like setup, right? Because we don't know if all the things we have investigated now, if they are just a construct of farmed animals, right? We know that the mycoplasma is also present in wild animals. There has been investigations on that already. No one knows really what it does, because mycoplasma is in general thought as the weirdo kid in the class, I would say, of bacteria. It's normally pathogenic, but there's no evidence of mycoplasma being pathogenic in salmonids at all. We haven't seen that in our data, and there's nothing published on that. So for me, it would be to like look into how mycoplasma is distributed in wild Atlantic salmon as a case, and maybe also look into, now we talking a lot about the bacteria, but since this diversity of bacteria seems to be quite low in salmon, I really want to know why is that? Is there a huge like host selection pressure? Like, is the salmon doesn't just want to keep everything out because it doesn't need it, or is it because of like virus, or is it all these things? So that's my next journey, I guess.
Yet one more layer. So I had a chat in one of the episodes with Jayla, who added the layer of the tapeworms. So it was this Matryoshka doll of the host and the parasites, and then the microbiome within each one. And now there's also, of course, in addition to bacteria and fungi and all that, a virus, and then there's bacteriophages and virus that are for the bacteria, and virus who are for the host, and so much to be discovered there as well.
It's very unknown ground, I must admit, and especially also the Jayla case, like with, I like to call it a babushka study, because it's like a host inside a host, or bacteria inside a host, right? And how this interplays, and it's quite intriguing, I must admit. And if we add virus on top of all that, everything starts to become really difficult, I would guess, or hard to explain at least. But
Yeah. So I look forward to hearing more from you in the future and in your future exciting studies to
Thank
take
you.
things forward and add more layers. You're a good bioinformatician, so you can deal, because every time, just for people to understand, every time we talk about an omic layer or different species, or it means more data and more complex data. And then you need to know, deconstruct things and know what's what, and then put things together again and model and try to see what's going on. And then depending on how you make your model, you end up with different results. And then you have to go back and check which one is the most likely, and is this real, or is it just in the computer? But then, and also as you highlighted, this is all in the setting mostly of aquaculture, which is very different from the wild. So is it the same? Is it not the same? But of course, then it has its utility, because it is a whole industry. So even if it's not the same, then having the results for aquaculture will definitely help the industry. But it's obviously then from an ecology and other points of view, nice to know what is happening in the wild as well.
Totally. Yeah. But
So,
yeah, and
yeah, go ahead.
yeah, well, as you said, like every time we add a layer of data, then we also add a layer of methods that no one has actually learned, at least in our department, right? And that's also like, it can also be quite learnful. I would say in this research field and in general in academia and research, like the learning curve is always steep, right? So which is nice, but
Yeah. And in the past, when we had simple data sets, it was a given that we knew how to analyze them. You just do ANOVA or you do whatever PCA. And so you know that you have the data, you put it in this program, you click the button, and then you get a cluster or you get whatever. But right now, and I think this is kind of a little bit of a hidden science, of course, not for the people who work on it, but for the rest of the world, people are coming up now with the methods, the computer methods and the mathematical and all these things, because they don't exist, and we have such complex data sets that it's not a given that we are going to be able to analyze them, or we are going to be able to extract all the information that they, or unlock all the information that they hold, because we might not have the method yet to do that. So yeah, it's very interesting also to see. One day maybe I'll attempt to do an episode about bioinformatics and see how that goes and how we can make it simple. Something that is absolutely not simple.
I would love to see that. So it would be great. Yeah.
Yeah, so it's nowadays it is, and so there we in the department we have people like you who actually do the lab work and then also the bioinformatics, but in some other cases we have separate people doing this, because both of them require a lot of knowledge and very specialized knowledge. So sometimes we have we divide the work in different people, and so we have people doing the lab work, and then once there's data produced, then it goes on to the bioinformatician, who then does the analysis and comes up with the pretty images that hopefully make sense out of the huge amount of data that is generated, and allow the scientific knowledge to advance.
Yeah, it's always a difficult thing that a lot of these data layers and in general all these methodologies, like a lot of them are really, really difficult, like both in the lab but also analytically and bioinformatics, right? So it might change hands quite a lot of times, and then actually doing the biologically inferring of the data in the end can be extremely difficult, because there are like small black holes, black boxes throughout the process, right? For the person in the end. which can be like essential for you as a biologist to actually know, right?
Definitely.
So that's also like one of the drawbacks at the moment, I would say. But at least like bioinformatics and all the analysis, a lot of very clever people are trying to make it more simple, easier for the general researcher, general biologist to actually do. So it's possible to like do the full circle yourself, I would say, which I also think is something this research area will benefit from.
Yeah.
At some point.
And it will be always and ever evolving. And one thing that you touched briefly upon is also that nowadays we need this to be multidisciplinary, because the interpretation, there's a lot of computing things that can be done, and models and comparison to databases and whatnot. And so the model can come up with things. But then there's the human, for now, interpretation of that. What does it mean when something goes up or down, or when A and B are associated in this context, but then B is associated with C in a different context? And that is usually in the light of current knowledge in the world, because maybe some things we don't know yet and we will know in 5 years from now, but also the actual people doing the analysis. Maybe there's something they don't know that other people know, or they're just being biased because they are neurobiologists or field ecologists, and so the interpretation is going to be different as well.
Totally. And like luckily you are never like alone in the research study. Sometimes you are. I would say it's not the normal thing to be like a single author, right, on the things we normally publish, right? And like, because I'm definitely not a chemist, I'm a geneticist or a molecular biologist, but definitely not a chemist. And I'm the same, I'm not an immunologist at all. These are extremely complex things, and we all need help to understand these really complex matters, because like metabolomics were very difficult. It's very difficult, at least to me, to analyze and investigate, because it can be anything. And and as you say, what does it even mean that it's up or down, like this specific feature? What does it mean for the host or for the bacteria? All these things we don't know at all, right? It's totally untouched realm again. So there's so much needed to be investigated before we have totally solid conclusions, I would say.
Yeah.
So, but at least we all the time see new sparks of associations, or like what can we say? Like it's too much to be improbable. So some of it simply must be real at some point.
Let's hope so.
Yeah.
Yeah, and in the context of our center, it's great to see that we have a lot of people with different and complementary expertise, and also we've seen the progress that to obtain certain data, the technology also needs to allow for that, and then we need an evolution also in the analytic and the analysis methods for all of that. And now we have Zoom in the internet, which means many years ago, people would discover things sometimes in parallel, and they've never seen each other, and they didn't even know about the other person's work. And nowadays we are so connected, we can just easily have a chat on Zoom with someone somewhere, or just send them an email, or even go to their lab for a short period. So it's things are looking up.
Definitely. So yeah, the interdisciplinarity would only be easier and bigger, most likely, because a lot of the questions we ask needs an interdisciplinary answer, I would say. So yeah,
definitely.
yeah.
So good luck with your future endeavors, because going to interdisciplinarity, maybe now you can find new friends and colleagues in the area of viruses and so on.
Yeah, that will be the next realm. Virus and in general, like the whole thing about this mycoplasma and salmon is it's so strikingly weird that it needs to be investigated more, because
but at least you've made the initial observation that it's weird, and now let's figure out why. What's up? Because it seems to be working out somehow.
Yeah, it's the first hint as a biologist, like this is too weird. Like, so yeah, I would look forward to that. But thank you so much for having me. Like, it was great to have a talk with you about all this, and I would always like to talk more about it. If you need more.
Thank you so much for your time and for a little bit of your brain, because it was a really, really nice chat. And it was, it started as a journal club. I think this was also kind of the idea with this episode, that we kind of introduce what is the scientific paper format, and then we go into actual papers, but then we just have a chat, which is kind of the main concept of this podcast, is that there is no script. I have notes that I cannot read because my handwriting is terrible, so I don't use them anyway. And then we just have a chat and see where things go within the context of the work we do and the remit of the Center for Evolutionary Hologenomics.
That's great. That's great.
Yeah. So thank you so much, and have a great day.
You too.
And good luck with your future endeavors.
I like the word endeavors for this kind of thing. I'll go back to my computer and work now.
Yes, on your computer endeavors. There you go.
Yeah. Thank you.
Thank you.